this is really the future, because that's really what I believe, and I try to convince as many people about this as I can. And yes, there are a lot of practical issues. I really hope that in 5 years all of the medical laboratories will be moving to this technology, because it really has a lot of advantages for our work, but also for the patients that we are trying to help. How far can we go with sharing data? Sharing data has huge advantages for patients, because we can use other people's expertise, other people's methods, insights to help resolve our patients, but then it is still private patient information. How far can we go in sharing that? But the most important aspect, I think, of our research is that we are showing to other labs what is possible and what we can do, and setting flag out there and saying, this is all possible. These are the benefits of technology. This is where we should be going. And my group is really working hard to get this technique really as a diagnostic test, which takes a lot more effort than you would think. Yes, we have it running up in research and we can do experiments there, but getting it in the diagnostics arena is different. My career has been a little bit of a coincidence, let's say. Because I did computer science. I didn't know much about genetics, but I ended up in genetics through an internship, and then I just stayed along. And I think I was very lucky at that time that just when I stayed, this next-gen sequencing technology came about. And with this technique, bioinformatics became really very important, much more important in genetics than it used to be. So the timing was very good. And then from there onwards, I stayed because I've always been very interested in trying to do let's say new stuff. So I work in a lab that does a lot of technological innovation. So we're always looking into new techniques to improve our patient diagnostics. And also there's enough room to do some more curiosity-driven science, let's say. I think the genetics has a nice combination. The fact that it's very applied in many ways. I work in a medical laboratory also, so they really do patient diagnostics. I think that's very, very nice. You can actually see the impact of your work immediately. Also, genetics itself is a very fast developing and novel field, and that makes it very interesting.
What led to the interest in working on this line of research?
Yeah. I think the main driver here in our lab is that we want to improve our patient diagnostics, and we do that in many, many different ways. But one of them, for sure, a big one, is through trying to implement the latest technologies as diagnostics. And we started already with long-read sequencing quite early in 2018, I think, also with some smaller papers, but these were more technological proof of concepts. Can we get data out of the machine? And because at that time we already envisioned the potential of the technology, and that slowly progressed over the years with larger studies looking at undiagnosed patients. And I think this last paper was really the pinnacle, let's say, where we could finally apply the technique at a much larger scale, where you can do more interesting studies. And I think the goal here was to finally also resolve this long-standing question. There's lots of papers out that show that you can find with this technique more variation, structural variation repeats, but the application to medical genetics, really looking at variants that are relevant to patient diagnostics, that had not been done. And I think this was the goal for this study, to show really in practice that it will have benefits in medical genetics to start using long-read sequencing.
Is this unique to rare diseases, or it can be applied to several other conditions?
I think our lab, we really specialize in rare diseases, but obviously cataloging as much genetic variation as you can is important for all kinds of diseases and all kinds of phenotypes. So I have no doubt that when costs start to drop, also for other diseases, people will start to look into this technology. I think the UK Biobank is already starting. All of us in the US is also already applying this. So I think the technique by itself is really giving us a much better picture of the variation that happens in our genome that was previously inaccessible, and therefore I think it will be relevant across genetics or for any kind of disorder. But the immediate, let's say the low-hanging fruit for this technology, I think could really be Mendelian disorders, like rare disorders.
So talk me through this technique. So what does it entail, and how is it being applied?
So the main difference is read length. So the standard currently is massively parallel sequencing, next-gen sequencing, or also sometimes called short-read sequencing, depending on the context. And it's defined by the fact that we can read the DNA in small pieces, like 100, 150 bases at a time. But then we do that massively parallel. We do it on massive scale. So we read millions or even billions of small DNA pieces together. And then when we have these pieces, from that puzzle back and reconstruct the original genome. Now, it turns out that actually reconstructing the genome from very small pieces is very challenging. And we cannot complete the puzzle. We can get to 95%, let's say, but that's about it. And I think here this new technology comes in. With this technique, long read sequencing, we can read DNA pieces that are much, much longer. In our case, we used 15 to 20 kb, 15,000 to 20,000 base pair fragments. And with that, we can make a much more complete picture of the genome. So we also get into this 5% that we miss, but also it allows us to better look at regions that are very repetitive or to detect larger changes in the genome structural variation. We know that we can pick up structural variation with short reads, but usually we sort of infer that the structural variant is happening. We look at coverage and say, oh, we see a coverage drop, that's probably a deletion. Or we say, oh, these reads, they don't align really properly, and maybe that's a duplication. But with long reads, because they are so long, they usually encapsulate a complete event. And because of that, we can detect much more of these variants in the genome. Several studies have shown that, we can get up to 3 to 5 times more structural variation cataloged in a human genome through this technology. So I think that's really the strength of the technique. And I think that was already clear, maybe even from the onset. I think what has changed in the last few years for this technique is that now it has become sufficiently accurate to compete with short reads. I think accuracy has always been a challenge for the technique, and that has been largely resolved. I think the pricing, the cost for doing such an experiment has become much, much cheaper. It's also becoming close to short reads maybe. And the throughput, we can do this now on a much larger scale, and that's what you would need for medical applications. And we can sequence now 1,000 samples in half a year, let's say, if we have sufficient machines. Previously, you could do maybe 2 samples per week or something.
There are different approaches to making diagnosis. We have the genetic approach, we have the immunologic approach, and we have other approaches. Looking at the way that attention is on genomics, do you think, in spite of the fact that, like you said, the cost is high, but it's coming down, do you think the future of reliable diagnostics for rare diseases is genomics?
Well, I only view things from a genomics perspective because that's my field. So for the work that we do, yes, a lot of that is genomics. I think genomics will always be, let's say, the first stop for rare diseases. Obviously, we are also seeing that even with these long read technologies, we are not resolving every single patient that we get. So there are still definitely challenges out there. And for this, we are not clear. So it could be that we're missing one big piece, but I think it's more likely that we're missing all kinds of small pieces there. And that to solve these patients, we may really need to look much more beyond genomics to resolve them, and also look into our transcriptome, our proteome, our metabolome, our epigenome. These kinds of things may be needed to resolve these for rare diseases.
In your study, I'm looking at it from a research perspective
Yep.
now. Do you think there is any specific rare disease that benefit the most from this whole genome sequencing approach?
I think every rare disease will benefit from this approach. I think that's also what we're seeing in some of the other studies that we're doing. If you ask me, like, where would you apply it first? I've always had an interest in neurodevelopmental disorders, so rare neurodevelopmental disorders, and I think there, because it is a very genetically heterogeneous disease, and we're still not resolving a large proportion of that. We know that most of the variation there that occurs that is causative is typically de novo variations, mutations that occur in the sperm or egg of the father and mother, so the parents are not affected, and it really occurs only in the affected child. And that makes it easier to resolve them in a way, because we can focus on these types of de novo events, and therefore it's a very good approach when you're looking at a new technology where you need to interpret like lots of variants that you find, especially of course with long reads we find even more. So where do you start? That can be very challenging. But for neurodevelopmental disorders, you could start with looking at de novo variation, and that might already give you a lot of information on how to do interpretation also in single cases. I think there the benefit might be the most immediate.
How do you see these being applied for? How do you see these being integrated into patient management? How do you see this potentially impacting clinical management and treatment decisions?
I think for patients initially, they will see the difference in their diagnosis. They will get more diagnoses. We will be able to resolve more of the patients than we do now. Yeah, that will also have an immediate effect on the number of patients that we can potentially offer treatment to. These long reads are also being used for other applications, and I know that some of my colleagues here working on therapies, for example, are also using long reads to characterize their cell models, and I can see that also there the technology will have an advantage. But I think, for patients immediately, the effect will really just be the fact that we will be able to help more of them with the diagnosis.
WGS is getting really, really popular now. And I think studies like yours are taking it closer to application in clinical setting. So what do you still consider to be existing barriers to that full-fledged deployment of WGS in clinical settings for rare diseases?
I think one thing is that it's just early days, right? So we think we need time. One of the big potentials of this technology that you can also see from the paper is that in our lab we have lots of different tests up and running, help out with different types of genes, different types of variants that we need to characterize. And keeping all of these tests running is a lot of work, takes us a lot of money, and potentially with long reads we can replace all of these tests. So we can get rid of lots of different assays that we're still maintaining and just replace them by this. But that takes time because we need to validate it for each individual case. We need to show that this is better. So I think that's one major thing, but it's just a matter of time. If you think about other immediate things that stop us from implementing this, I think, we're not stopped for implementing this because we will this year actually start with offering the first diagnosis with this technology. But the challenges there, I think, are still definitely the pricing of the experiment compared to the existing short reads. It's still relatively expensive. I think it's the throughput. The machines, you need a lot of machines to get to the high numbers that you have in a typical diagnostic lab. And we would like to do maybe 20,000, 30,000 tests per year. I think that is, with the current technology, not yet really feasible. So we will have to start a little bit smaller. And otherwise, I think it is indeed the methodology that needs to be developed. So people are working hard on that, but getting the software there, getting the analysis methods, also the education here of our laboratory specialists to know how to deal with these type of data, how to interpret this. But that's just a matter of time. So I think from our perspective, I think we are definitely going there. And this year we'll have something up and running as a diagnostic test for sure. And I have little doubt that in 5 years, many other labs will have followed.
What does the diagnostic testing that is expected from your lab going to look like? And if you can give me an insight into the current cost implication so that I can make a comparison on the other options available.
We need to consider not just the consumable pricing, but we need to consider instruments. We need to consider lab personnel, additional equipment that are needed. So I can only give you a ballpark idea about what in my mind is the cost difference. I think long read sequencing is roughly twice as expensive still as short reads, and that needs to drop. But I think already big steps have been made in the last few years, and that's why we're able to do these studies now in the first place. That's also why we're able to do this year. We are planning to do 5,000 long read whole genomes, and I think that's because the cost is becoming competitive. But it's still more expensive. So I think for many labs, that's going to be a limiting factor.
I'm curious to gain your insights around this issue of variants of uncertain significance. For instance, let's say the clinician is presented with that information from an expert perspective in the laboratory. How do you expect this information to impact or guide patient counseling?
I think we will definitely not get rid of variants of uncertain significance with this technology. I do think it will help because we are already seeing that sometimes we find variants that we do not know are pathogenic, but we cannot do, for example, segregation analysis because parents are not available. In this case, with long read sequencing, we can do allele phasing. and then see that these variants maybe do come from different parents, and then in that way resolve variants of uncertain significance. I think we have definitely already seen the first examples of that. We've seen examples of, for example, a de novo mutation and an inherited mutation, and with the de novo mutation, we don't know from which parent it came. But then with the long reads, we can phase this and thereby resolve then this variant of uncertain significance. So that there the technology will at least help us. I think the bigger issue of variants of uncertain significance in general, I think, will still remain, and there we'll need to move to broader omics testing that also considers gene expression, metabolomics, epigenetics, these kinds of things for us to get better at interpreting these variants. So unfortunately, I think, many patients will still have to, and physicians actually will need to keep dealing with these variants of uncertain significance.
I know you said we are in the early days of BGS in rare diseases, but do you see it becoming a standard first-line diagnostic tool?
Absolutely. Yeah, absolutely. So I think it is early days, but we are moving at an, I think, extremely fast pace. And as I said, I think in 5 years, many labs will be using this technology. We've now last year seen, for us, we've been working with PacBio a lot. They've developed a new machine last year. I think if they do one more iteration, then the throughput and the pricing will be such that everybody can start using this. I think they already put a new machine on the market that is a little bit smaller, the Vega, for people to work with. I think for us, in our mind, but we have done, of course, a lot of studies already on long read sequencing, is absolutely clear what the benefits are of this technology and how amazing it is compared to our short read technology. So I think for us in 5 years, we will not be doing much short reads anymore. Everything will move to long reads. And in my mind, it will go relatively fast.
Interesting. So, with the insights you know about how to carry out the BGS in the context of rare diseases, I'm curious to know how do you think policies and regulations should evolve to support the BGS adoption while also minimizing the adoption risk?
With every new technology, rules and regulations need to be adapted. Also for us as a medical laboratory, we get financial reimbursements for tests as we do, but these tests we have to go to health insurance companies for this. And obviously with new techniques, we need to talk about these things again. So it is my hope really that we are a little bit better prepared than we were when next-gen sequencing came. I think there it took a long time for all of the ethical, legal, and financial issues to be arranged. I think we are more prepared for that. I hope also that governmental organizations and policymakers are better prepared. I think it will be easier than it was before, because we already have a lot in place for short read technologies, and then maybe long reads will just be a relatively modest step. But this is definitely something that we are also now working on to make sure that this goes smoothly, but will be a less big step than when we move to sequencing in the first place, I think. So hopefully it will be a lot smoother.
There's also a lot of talk around AI and machine learning in WGS rare diseases. It's just ambitious buzzwords that we are saying, or do you see these also plugging in?
I think it's, for sure, it is definitely a buzzword. I think everybody agrees on that part, but I think there is definitely some reality to that. I see a lot of laboratory specialists spending time on interpreting variants and trying to figure out whether something is relevant for disease. I see also in basic bioinformatics methods now that deep learning is becoming the standard for detecting variants. So there's clearly a lot of potential there, and a lot of things are actually already being used concerning this. So I think it will definitely have a big impact. Whether it will be as big as long read sequencing itself, I'm not sure. I think for that we may need a little bit more time. And of course, the rare diseases has the unique challenge that, they are rare. And there's a lot of niches, exceptions, and I think that makes it challenging for AI to make huge steps there. But also in this area, in my opinion, I think in 5 years, it might look very different. And I think everybody who will do genetic interpretation will be using the assistance of AI methods.
Okay, so what do you consider to be promising research areas or collaborations that are emerging in the field of WGS in rare diseases?
I think, we still have a little bit of the promise of multi-omics, and I hope that's also for us still an open question. How much is that going to add? How useful is it going to be? I think there also we are typically limited by the fact that we always get blood samples, let's say, so we're never able to look into the relevant tissue for most disorders for our multi-omics. But I hope there are also some new techniques on the horizon that allow us to do more work in relevant tissues. And so I hope the multi-omics is something that I hope will be the next layer on top of having a complete genome. So that for me is an exciting area of research.
So I'm curious to know your thoughts around equity and ensuring that every individual that needs access to this diagnostic tool could be able to access it. I know you are a tech person and a researcher, but are you personally thinking about this, or what are your thoughts on that?
Yeah, so I mean, there's lots of different kinds of equity, right? That one can consider. I think one of the aspects, at least that I do a lot, is I try to do a lot of education and teaching. So I organize lots of teaching courses around the world, also quite often in countries that have less financial resources, so that at least they have the knowledge to do the things that they want to do. And then they only have to worry about, let's say, the finances, but at least that the expertise level is the same as you would get, let's say, in other countries. So I think that in terms of equity is the only thing that I'm really actually involved in. But for sure, this is a big challenge. I hope, and we do see that, of course, next-gen sequencing initially was only for the privileged, and now it's become so standard, it's become so cheap, so easily accessible, that this is a problem that is slowly resolving itself in many ways. And I think then still the problem is then no longer technique or access to the technique. The problem then becomes having the right knowledge and expertise to use the technology. I think that's where I try to make a difference.
Are there any infrastructures that you still think are needed to fully maximize WGS?
I think the short reads is going fine. I think we have a lot of development has been done there in terms of infrastructure, and it's become really, I think, in many ways a very standard approach now. I think for the long reads, there is still a lot of work to do in method development to get everything that is in the data out of it. And yeah, I think most of the infrastructure that you would need would there really also come from companies. Right, really, the suppliers of this technology need to make their technology easier, faster, cheaper. I think these are the big things that need to happen. And as I said, I'm excited about the benchtop PacBio sequencer that is out now. I think this was one of the advantages for sure also of ONT technology, that it was a relatively easily accessible technology for many people, and that you could start playing with it. But now I think for diagnostics application, we need to have solid pieces of software. We need to have much more robust infrastructures in terms of analysis and interpretation tools. But that's what we are working on.
So what's next for your research team, and what are you guys doing there currently?
Oh yeah, my research team goes into many different directions. I think one of my favorite topics is still looking at de novo mutation and population variation as a very more fundamental genetic studies. And in other terms, really, my group is really working hard to get this technique, the long read sequencing, really as a diagnostic test, which takes a lot more effort than you would think. Yes, we have it running up in research and we can do experiments there, but getting it in the diagnostics arena is different. And then, we're also looking into interesting features of the technique, like methylation analysis that you can do from PacBio, and seeing if we can use that, for example, for episignature analysis, which is also a very promising method to deal with variants of uncertain significance. And this has been demonstrated for array technology, but it still needs to be set up and established for long read technology. I think that will be something very nice because you get it for free on long reads, and it will actually help a lot of patients. But there we still need to really develop the methods.
What potential impact do you think the research that you and your team does could have in rare diseases in general?
I mean, not necessarily just me, but I think a lot. I think what is the most important aspect, I think, of our research is that we are showing to other labs what is possible and what we can do, and setting really let's say a flag out there and saying, this is all possible. These are the benefits of technology. This is where we should be going. So I really hope that also with our paper now, other laboratories are seeing this and are thinking, okay, maybe we should also start moving into this field, because it is now actually diagnostics ready and it has really clear benefits compared to the current technologies that we're using.
What do you consider any possible ethical issues in handling UGS for rare diseases?
I have not encountered any issues where I think these are really ethical constraints. I think one of the things that we always run into in terms of ethical issues is how far can we go with sharing data. Sharing data has huge advantages for patients because we can use other people's expertise, other people's methods, insights to help resolve our patients, but then it is still private patient information. How far can we go in sharing that? I think this is for sure an outstanding issue that also many people are dealing with, and there is progress being made. We have large projects like Solver-D or Erdera in the EU that are tackling these issues and are making progress, but in daily practice, this I think is still an ethical issue that we need to really think about.
Thank you so much. So what are your last remarks for me?
Well, I hope I convince you that this paper and also the work therein, that this is really the future, because that's really what I believe, and I try to convince as many people about this as I can. I really think we should not be doing short reads anymore. We should really move to the long reads. And yes, there are a lot of practical issues, but as I said, I really hope that in 5 years, all of the medical laboratories will be moving to this technology, because it really has a lot of advantages for our work, but also for the patients that we are trying to help.
Thanks so much, Kristen. I really enjoyed talking to you.
Thank you. It was a pleasure. Very nice.